Good evening, everyone. We are here with yet another virtual session on our platform. Today, here we are with Dr. Heena Saini. She is currently working as Scientist C at All India Institute of Ayurveda, New Delhi. She has completed her BTech degree in biotechnology from Banasthali University, Rajasthan, and she has earned MTech degree in biotech as the first rank holder from Maharishi Markandeeshwar University, Haryana. She had her formal research training from Premier Institute in India, CSIR, in Delhi, and she has also worked as SR Fellow in CSIR in the year 2015, and she did her PhD at BITS Pilani, Rajasthan. During her work of PhD, she worked on protein homeostatic machinery autophagy to design a therapeutic strategy to combat cancer via targeting key proteins like mutant P53 and YAP1. She is awarded a senior research fellowship from CSIR and ICMR, and she is a member of international group which is Women in Autophagy, and she has more than 5 years of teaching experience in the field of teaching and in the field of cancer research. Thank you so much, ma'am, for your time on our platform. The platform is yours now.
Thank you so much, Dr. Bhavit, for your introduction. And first of all, I would like to congratulate OYG for starting up this platform and inviting me for today's talk. So, Dr. Bhavit already mentioned, I'm not a medico, but a scientist, and I did BTech and MTech. So, with my expertise in cancer and molecular biology, with all the little bit of knowledge that I have gained so far, today I will discuss the molecular understanding of cancer development in humans. So, as we all know, cancer is not new to us. We all are aware about these deadly diseases. After cardiovascular disease, cancer is the second most reason for death worldwide. So, as we look at the statistics, uh, my slides are not changing. Just a second. Okay. Okay, as we look at the statistics released by Globocan 2020, cancer bears the 19.3 million new cases, and there are 10 million cancer-related deaths worldwide. And as we look into these bar graphs, breast cancer is the topmost cancer, which bears the maximum number of cancer cases, which is 11.7%, followed by lung cancer, colorectal, and so on, while lung cancer bears the maximum number of cancer-related deaths in the world. And as we look at this pie graph, country-wise, Asian countries are responsible for majority of the cancer cases, where China stands at number one with 23.7% of total cancer burden, and India stands at number three with 6.9% of cancer cases. And it is estimated that by 2040, there would be a 50% rise in cancer cases. Of course, this is alarming. And so many researchers, medicos, clinicians, they are working so much to come up with better strategies to combat this disease. So let's talk about cancer. Let's try to understand what is cancer. As we all are aware that about this definition of cancer, that it is a disease of abnormal growth and proliferation of cells which further invade and destroys normal cells. So before understanding cancer, we should understand what is this abnormal growth and proliferation. So in human body, the smallest part is cell. So the cell is responsible for the whole functioning of the body. Whatever an organ is doing, organism is doing, it is coming up from that cell only. Okay, and this cell maintains the equilibrium. Like a cell grows, it does its function, and if it dies after a certain time when its job is done. Okay, and a cell is in normal scenario, a cell divides in two daughter cells, and if there is a full cell is formed, then it will do its function normally. But if there is a damaged cell is formed or there is a faulty protein present in the cell, it will first go for some kind of checking, whether the cellular machinery can rectify this fault or not. If that is not happening, then we will go for cell death, which is a programmed cell death, that is apoptosis. But in cancer, even when a damaged cell is formed, instead of going for cell death or apoptosis, these cells keep growing. Okay, if there is a mutation in a gene, in a protein, if there is a mutation, it will lead to another mutation that will lead to another mutation. This is known as driver mutation. Like we all have heard about the COVID. So first strain of COVID was something else, then it got mutated, it got another strain. Likewise, in cancer also we get multiple mutations, and these mutations make cancer cells more stringent. And these mutations help them for more proliferation and for more growth. And since the cancer cells are not going for cell death, they are immortal. So now why it happens with cancer cells, like how are they being immortal and how are they proliferating so much? It all depends upon the good protein or bad protein. So I will try to make it as simple as possible. Okay, so we'll talk about good DNA and bad DNA, good protein and bad protein. So here, good DNA and bad DNA means the DNA which is helping cancer to proliferate or to cause disease, to proliferate and to be immortal is the bad DNA, and the one which inhibits it is the good DNA. Okay, so this is the central dogma. DNA, which is the genetic material present in our body, which has the like key for everything which is going to be prepared in the cell, is there in the DNA. It gets transcribed to RNA, then it gets translated to protein. Okay, and then this protein is responsible for everything that is being reflected by the cell. So if there is a good protein is present, then obviously that will make us healthy. If there is a bad protein is prepared, then that will make us diseased. Okay, so cancer cells, these cells are very smart. Cancer cells are smart. So what they do, they try to make more of the proteins which help them in growth, which help them in proliferation, which help in angiogenesis, which help them to live more. Like they can prepare protein which inhibits the death signals. Okay, and they inhibit whatever is inhibiting their growth. Okay, so just look here. This is a simple diagram of cell. Okay, so cell has a membrane. This membrane has receptors. These receptors collects the signals. So whenever a signal comes and binds to this receptor, these receptor gets activated, and they activate further proteins or enzymes inside the cell, which further activate other protein enzymes. Okay, so this is a cascade of reaction which happens. Like first thing gets activated first, then it will activate another, it will activate another, like that. Okay, so once that particular protein is activated, it will go to the nucleus. This is the nucleus of the cell where DNA is present. DNA contains the genes for several RNAs, and which is going to be protein further. Okay, so a protein goes into the nucleus. It will go and bind to that specific gene, and it will result in the transcription of that gene. So mRNA is formed. This mRNA will form the protein, and which is responsible for the cell functioning. Okay, so this kind of reaction happens for all, right? But there would be a different signal marker, there would be a different receptor, there would be a different protein, and there would be a different target gene. And that is how different proteins are formed, different genes are getting transcribed. So cancer cells chooses which protein to be formed more. And like I also mentioned about the mutation, if there is a mutation in this target gene, then of course the protein which is going to be formed is not the ideal one, but the mutated one. And of course, the mutated proteins have different functions. Okay, and cancer cells also play not just with the formation of these proteins, but yes, with these intermediate molecules also, like these receptors can also get mutated, these signals can also be disturbed, these proteins can also be disturbed. Okay, so likewise, cancer cells facilitate the growth of or the formation of those proteins which help them their own growth, and they inhibit those proteins which inhibits their own growth. Okay. Now coming to the basic characteristics and classification of cancer, like I said, they disobey the growth control, and that's how they proliferate more. They escape the death signals. That's why they are immortal. They lose, now they don't have any specific function. They lose the property of differentiation. cancer cells have no function. Okay, actually they have one function. They take the nutrients from the normal cell. So cancer cells, what they do, because they need more energy, more growth. So what they do, they start taking the nutrients from the adjacent normal cell. Okay, and these are unstable. They are always prone for newer mutations. Okay, they overrun their neighboring tissues and invade. Yes, invasion and metastasis is an important property of cancer cells. Like if you see this diagram, this is a skin. These are skin cells which are well arranged, but when there is a change in one cell, so this cell will start growing rapidly. They start proliferating, and there is an increased number and size of this single cell only, and then they start invading. They start invading from their own territory and starts going to the territory of the adjacent cells. Okay, this is the invasion, and then they enter the bloodstream or lymph vessels, and they metastasize to distant places, and they cause cancer there. Okay, these are the hallmarks of cancer. Okay, like I have already discussed that cancer cells are very smart. They accelerates the protein which help them in grow, and inhibits the proteins which inhibits their growth. So that explains the hallmarks of the cancer. Like they evade growth suppressions, like whatever suppresses the growth of cancer cells, they evade those pathways. Okay, very smart. They have their own soldiers to fight those proteins which inhibits its growth. They avoid immune system. Of course, immune system recognize the foreign protein. Okay, so if there is a change in protein, immune system can attack that, but they are very smart. They avoid immune system also. Likewise, they promote tumor inflammation. They induce invasion metastasis. They promote angiogenesis. There is a cell death. They sustain proliferating signals. So these all properties are governed by cancer cells by playing with genetics of human body. Now, what are the causes of cancer? Like we all have heard about different causes of cancers, like UV rays cause the cancer, different mutagens and chemical carcinogens are there, like some benzene compounds, they cause cancer. There are several viruses like papilloma virus and hepatitis virus which cause cancer. So I would say these all are the external factors, but whatever triggers the cancer in the body, whatever could be the causes among the ones listed here, the main cause is genetic instability. So in our body, we have certain genes. For cancer, we have two major genes, oncogenes and tumor suppressor genes. There is an equilibrium. There is equilibrium maintained in the cell, which is responsible for the proper function of the cell. Okay, so tumor suppressors the proteins which prevents the cell growth, while oncogenes are the proteins which promotes cell growth. So let us understand this like this. So we are in a car, and if we have to stop a car, we'll put the brake, and if we have to speed up, we will put the accelerator. So tumor suppressors work as car brakes, and oncogenes work as car accelerators. And most of the tumor suppressors loses their function when there is cancer, because tumor suppressors will suppress the growth of cancer cells, and once they loses their function, they are not no more available to suppress the cancer growth. While oncogenes, they are gain of function. In our body, there are proto-oncogenes present, but they require some kind of signal which will activate them. So once they get activated, they get converted to oncogenes, they acquire new function. Okay, and once they acquire new function, now they are ready to cause cancer. Okay, so and one more thing, these tumor suppressors, they can also work as oncogenes if there are two hits. I will discuss that in my further slides. So let's discuss about one of the famous proteins in cancer, which is P53. So P53 is a protein which is a tumor suppressor protein. It suppresses the growth of cancer cells. Okay, so like I said initially, that if there is a faulty protein is formed, a faulty DNA is there, a damaged cell is there, so P53 is responsible to induce apoptosis. Okay, that is how it suppresses the tumor growth. But in more than 50% of cancers, this protein gets mutated. And once this gets mutated, it becomes oncogenic. Like when the protein is present in its wild type form, which is a tumor suppressor form, okay, it will go and bind to the DNA. It will act as a transcription factor. It will go and bind to the DNA, and it regulates the transcription of genes which are involved in apoptosis, growth arrest, or senescence. All are the one which controls the growth of the cell. Okay, but when this protein gets mutated, they no more, it does not bind to the target, which will lead to the transcription of genes for apoptosis, growth arrest, and senescence, but it will bind to a new target, which will lead to the transcription of genes responsible for chemoresistance, proliferation, and other oncogenic genes. Okay. Like when this protein has acquired gain of function, now it has gained a function which was initially absent in its wild type form, which was a tumor suppressor form. Now it has gained its function, which is oncogenic function, by binding to newer proteins, by binding to new promoters, binding to new transcription factors, and by inhibiting new proteins. So basically, whatever it was doing initially as a tumor suppressor, that activity has changed and it has acquired mutation. Uh, we have heard of this Romani syndrome. This is a rare disorder which increases the risk of developing several cancer. Okay, so this is a disease. But why this disease happens? Because it is associated with a germline mutation in P53 gene. So many children and adults who have germline mutation in P53, they are prone to this Romani syndrome. Okay, and many generation of one family, if they have this genetic mutation in them, then many generations get cancer disease. Okay, so this Lee-Romani syndrome includes different types of cancers like breast cancer, sarcomas, leukemias, and many more. Another example is BRCA gene. BRCA is breast cancer gene. It is also a tumor suppressor gene, but when gets mutated, it can also cause cancer. So BRCA gene is responsible for breast cancer and ovarian cancer. And if a woman has BRCA1 or BRCA2, any of the gene with mutation, then she has 5 times higher risk of breast cancer and 10 to 30 times higher risk of ovarian cancer. BRCA gene got very famous in 2013 when Angelina Jolie, she decided to to cut out her breast. She went for mastectomy. Why? Because her mother died of cancer, ovarian cancer, because she had BRCA1 gene mutation. And when Angelina Jolie, she also found that she has the same mutation. Then she went for mastectomy, and in her article to New York Times, she said that now she has dropped her chances of breast cancer by 87 from 87% to under 5%. Like tumor suppressor genes, our body has oncogenes as well. Um, so a very simple diagram of the cell, there is a growth factor, or I would say a signal which will go and bind to the receptor, which will activate the downstream signaling enzymes or proteins, which will activate the transcription factor, and hence the transcription occurs. So which will lead to cell proliferation, cell apoptosis, DNA repair, or anything. Okay, so these oncogenes, they can target any of these component of the cell, uh, and that facilitates their growth. Like EGFR, HER2, these are the receptors. Okay, if these receptors are mutated, what happens? They act like as an on switch. So they keep sending the signals for the downstream proteins to prepare the protein of their choice. Okay, if like I'll say there is a protein which is responsible for growth. Okay, so the transcription of that particular gene happens, but if the receptor is mutated, it will not stop that, but it will try to keep making it will try to keep sending the signals to make more of that gene, to make more of to transcribe more of that gene and to make more of that protein. Okay, and there nowadays targeted therapy is already available, Like for EGFR in lung cancer, people use erlotinib as a drug to target EGFR. For HER2, which is there in breast cancer, people are using trastuzumab, which is an antibody-based medicine. Okay, now there are different regulatory GDPases. These GDPases are nothing. You can understand it like these are the facilitators which help in the signaling cascades like RAS proteins, and there are different transcription factors like MYC genes. So these all are oncogenes. They are not functioning properly because they all are involved in proliferation. Okay, most of the ones which are discussed here are the ones involved in proliferation. Likewise, there are oncogenes which are responsible for inhibition of death pathways. Okay, like transcriptional activators, YAP. So in proto, these are the genes which are responsible for the uh, growth and proliferation. But if they are not functioning properly, then there would be an uncontrolled growth and proliferation, which will lead to cancer. That's why they are known as oncogenes. Okay, so how can we identify these molecular changes? Like if a person has a cancer or not. So for that, what we can do? We can check for the gene we can first do the gene expression. Like most of the cancers have certain markers, so we can check the expression of these markers. So, like I'll take a cell from a healthy lady for breast cells, and I'll take a breast cancer cell, and I want to compare the expression of suppose HER2. Okay, then HER2 expression should have come more in the women of breast cancer. So likewise, we can check the gene expression by amplifying that particular gene using PCR machine, and like shown here in this, that particular cancer marker will show more expression in cancer. We can also do the protein expression studies. Protein expression studies can be done using immunofluorescence, using microscopy, or immunoblotting, which is famously known as Western blotting. So immunofluorescence can tell you the localization of the protein, it can tell you the concentration of the protein, whether it has increased or decreased after the disease. And likewise, we can quantify the protein using immunoblotting as well. Okay, so and yes, there are certain assays also by which we can check the presence of a molecular marker. But if there is no molecular marker available, okay, how can we check how can you identify these molecular changes? We can go for sequencing. Okay, so you can take the sample of the diseased person and you can sequence the genes. Then we can compare it from with the normal cell, like what is the expression of a particular gene in normal cell and how it is changing in cancerous cells. Okay, let's discuss molecular understanding of cellular death. So how do we, these are like, lately I just discussed about the markers of the molecular markers, but how do we do it in research? If I have to study, there is a drug. Okay, you people like might be interesting in knowing if you have a drug, how would you check whether this drug is inducing death, whether it is inducing proliferation or not? That's why I have included few slides here. Okay, so if I have a drug and I want to check whether it is inducing death or not. Okay, so first I want to check apoptosis. So apoptosis is a mechanism which is a programmed cell death. What happens in apoptosis? Like we all know that cell membrane has phospholipid over it, and during apoptosis, this plasma membrane, it gets flipped out, and the phospholipids which were hiding inside, they are now available outside. So what happens? This annexin 5 has a tendency to bind with this phosphatidyl serine and pi dye that I have used. So during apoptosis, when this phospholipids are available, this annexin 5 will bind to it, and this annexin 5 is conjugated with FITC, which is a fluorescent compound. So more the apoptosis, more is the binding, more would be the fluorescence of FITC. And pi is the stain used to stain the dead cells. When cells get when cells die, they become more porous. So this pi can enter inside the cell and it can bind the DNA. So if there is more of dead cell, there would be more of porosity. Pi can penetrate inside the cell, and it can bind to the dead cells. So this is the basic principle of an exin 5 PI assay. So how do we do it? We do it using the flow cytometry technique. Okay, so the data is represented in this four quadrants. So in x-axis, I have taken FITC and exin 5. In y-axis, there is rubidium iodide. So this first quadrant is for 0 FITC, 0 PI means viable cells. And if cells are in early apoptosis, because in early apoptosis, porosity is not there. So early apoptosis is an exin 5 positive cells only, which is this. So if I have to calculate about the number of cells in early apoptosis, I will calculate this quadrant, which is FITC positive only, but not PI positive. Okay, in late stage, when an exin can bind to phospholipids, and PI enters this. So this is apoptosis positive, and these are necrotic cells also. So they can come here in late apoptosis. Okay, so if I have to count the late apoptotic cells, I will count this quadrant, which is annexin and PI positive. And if I have to count the cells which are ultimately died, then I will count this quadrant, which counts the necrotic cell population, which is only PI positive, because all the cells died here. Okay, likewise, you can compare with, like, you have taken, suppose this is the control, this is drug 1, this is drug 2. As you can see, there is more number of cells here, here, and here in comparison to here. So likewise, you can compare whether your drug is inducing death or not. And then you can calculate the percentage. If I have to calculate the total dead cells, I will calculate this, which will give you one number. And then likewise, you will calculate for other concentrations also. So you can say, like, with 100 micromolar of any of the drug that I have used, there is significant cell death. Okay, so apoptosis is a pathway. I will not explain the pathway. So like other pathways in apoptosis, there are several proteins which plays their role to ultimately make a cell go for apoptosis. Caspase 3 and PARP are one of the important, like I'm showing the data of these two only. So these are one of the important proteins which plays important role in apoptosis. So we can check the activity of caspase 3. If there is more activity of caspase 3, then whether this drug is inducing apoptosis or not, then of course we can check using caspase 3 enzyme activity. Its activity is increasing means it will lead to more of apoptosis. Okay, we can check for protein markers, like here we have checked for cleaved PARP, because PARP gets cleaved before apoptosis. So there is more of cleaved PARP. It means the drug that I have used, it has increased the number of dead cells that we have checked in previous slides. It has increased the activity of caspase 3. It has increased the cleaved PARP also. It means the drug that I have used is inducing apoptosis. Okay, there are some of the proteins mentioned here, like BAX and BAC, BCL2. So there are several anti-apoptotic and pro-apoptotic proteins also. You can, they are the markers of apoptosis. So you can check the expression of these proteins also, like BAX and BAC, these are pro-apoptotic protein, while BCL2 is an anti-apoptotic gene. So you can check them on genetic level by doing the PCR, and you can check them on protein level also by doing the Western blotting. Likewise, if I have to check the cell proliferation, whether my drug is inducing proliferation or not, we can go ahead with MTT assay. Okay, that that actually calculates the number of viable cell inside the cell. So if this is the control to which I have not given any drug, then if you have given drug 1 and drug 2, there is more number of cells after a particular duration of time. So that tells us that there is a cell proliferation happening. Okay, so these experiments that I have explained, they are on the basis of cell culture. Okay, we can grow the cells in culture, human cells in culture, and then we can do such experiments. Okay, such experiments can also be validated through patients' blood samples also. Like you can isolate RNA, or you can isolate protein, and you can go ahead with gene expression and protein expression profiling from patient samples also. So wound healing assay, like suppose this is a plate which has many cells. So I have made a scratch, and these dots are the cells, and this scratch is the area over which no cell is growing. Then I have given drug D1, D2, D3, and I have kept it for some time. And as you can see, sorry, this D1, D2, D3 here represents the different times. Okay, suppose I have observed it after 24 hours. This scratch has shortened. Okay, and after 48 hours, it has shortened even more. And after D3, it is very small space. Okay, it means cells are proliferating, they are growing, they are migrating. Okay, so wound healing assay also confirms the proliferation. And there are certain markers in our body like Ki67. It is a well-known marker for proliferation. So in cancer cells, we also check for the expression of Ki67. If they are showing there is a higher bar in cancer, it means there is more expression of Ki67 in it. So Ki67, it goes to the nucleus, and it is responsible for the more formation of ribosomes, which is required for uh, protein synthesis. Okay, and this is a biological pathway, multi-biological pathways actually, which lead to proliferation. Like I explained you, there were different receptors and there were signals. So you just assume for WNT, there are different receptors. Here for PDGF, there would be a receptor here like that, and they activate the downstream proteins, like this activate this. And believe me, this is not as simple as it looks here. There are many proteins or inhibitors or activators involved in these pathways. So ultimately they transcribes the genes at the end, which is responsible for proliferation. So apart from checking this, we can also check the expression of these genes or proteins to confirm the proliferation. If the expression of like c-Myc is increasing, I will say that proliferation is happening. If cyclin D1 is increasing, I would say proliferation is happening. Okay, but yes, one thing that we have to keep in mind, that proliferation is not the only function that these genes or proteins are doing. They are involved in multiple functions. Okay, so that is complex that we are not going to discuss in today's talk. Okay, there are many treatment options available for cancer: surgery, chemotherapy, radiotherapy, immunotherapy. But yes, there's many hurdles in them because we have less knowledge of the molecular mechanism. That's why there is a new therapy which has been introduced, which is targeted therapy. But in targeted therapy also, we are targeting one or two genes. But like I said, this that like these molecular mechanisms are very complex. So there is a requirement for better understanding of these molecular pathways. Okay, then only we can get good results with all these therapies. So, though we have these therapies as a conventional method to treat cancer, but there are major hurdles as well, like chemo resistance, lack of effectiveness, side effects, morbidity. And how we can come up with this major hurdles? By doing the advanced research, which is the need of the hour for every type of medicines available in the world. Because if we'll do the research, then only we can come up with the better strategies, and then we can come up with the personalized treatment. Yes, that is required because each and every cancer is different for different patients. Then we have, okay, like I talked about personalized medicine. So I want to introduce this term integrative medicine also. Integrative medicine means when we are taking different types of medications together, like conventional medicine, which is already there, known as Western medicine or modern medicine. With that, we use the traditional medicine, which is most prevalent in the Asian countries, and the therapies which involves the lifestyle intervention for the holistic approach. With this, I conclude. I think I have a very less time. Okay, with this, I conclude my presentation. Like I said, there different signals activate different pathways. Like this red one is for metabolism, changes in shape, new proteins. So whatever happening inside the cell, it's because of this molecular mechanism which takes place. And this is not this simple. Like I said, each molecule, each protein is involved in multiple functions. So like this protein is involved in the activation of this, inhibition of this, and it is even more complex than that, even more complex than that. So at the end, I would say people call it fate. We know it's genes. And I also want to conclude with this. With conventional method, we need traditional medicine, a better lifestyle, and research. Then only we can prevent and cure cancer. Thank you so much.
For the wonderful presentation what we had in the last 30 minutes. So my next question would be that how do we think that the research progress in the field of cancer, or how we should progress ourselves as a doctor with our academic career, how we should proceed in cancer research?
Okay, like I said, we have done we have got a lot today with advancement in different techniques. We have reached a certain milestone, but the disease has not been cured yet. So basically, for that there is an understanding of this. Like I said, more of research is required. There is a better understanding of this. Like I said, there is one gene which is responsible for this disease. That cannot happen because there are certain other genes also which is not known. So as a doctor, I would say that uh, for cancer, we cannot just treat patients with whatever resources we have. Side by side, we have to do the research also. Okay, so we can come up with the personalized medicine. We can design the treatment basis on the basis of the patient's requirement. Okay, so I think that is necessary.
What are the hurdles in your research you are facing in terms of integrative medicine or integrative approach in the research in terms of cancer?
Okay, actually, this is very complex. You might feel that most of my answers are like centering towards one thing only, because in research, what happens, like I said, okay, I'm doing a research on, I have taken, suppose, lung cancer, and I have taken a drug, and I am seeing that, okay, this drug is giving me good result. This is inducing apoptosis also, this is inhibiting proliferation also, but later on, you'll come up with a certain gene. Okay, you were focusing on one pathway. Okay, and then you come up with another gene or another pathway which is getting upregulated, because there are like thousands of pathways and genes and proteins in this inside the cell. So you are targeting one, you're focusing on one, but there are others also which are involved. So you will see that people who are doing with research on cancer, so some people are working on EMT, epithelial to mesenchymal transition. Some people are doing working on epigenetics. Some people are working on protein degradation machineries like autophagy. Some people are working on telomerase. So there are like so many things for cancer research, and there are so many factors responsible for cancer. Okay, and the whole thing which is playing is genes. So we are still looking for that center gene which we can target to come up with good strategy to combat the disease.
And it's a convincing answer. It's not like repeating in a circle for me also. Uh, so my last question would be, what would be your guidance or your points, takeover points to all of us if we wanted ourselves to target in research in terms of cancer or in the field of cancer?
Okay, so we give medicine, like somebody has prescribed, somebody has said that this is the particular medicine for cancer. Okay, so but we all are aware that we have not come up with the treatment which can completely cure it. The biggest hurdle that I have seen in cancer is the recurrence of cancer. Okay, why they reoccur? Because that gene is still present. Okay, there is a tiniest minute part of cancer is still there, which was not removed surgically, which was not removed by chemotherapy, because either it has metastasized to some other part which was not because once it has metastasized and we did the PET scan or something, we did not observe it, because initially that damage was in a single cell, but with time it proliferates. Okay, so that is I suggest that not just uh, targeting cancer at once. We of course we do follow-up studies also, because that is required. And the major hurdle I was talking about was the chemo resistance, because these cells, they might show death for certain times, but they remain as persisters. Okay, they are persisting the growth in drug environment also, they are not dying. So basically, not just treating the patients with drugs because somebody has prescribed it, this will not solve the concern. So because we have more medicos here in our country than the researchers. I would suggest with the medical or clinically, whatever you are observing, you must also do the research side by side. Because of course, there would be certain other targets which we miss out when we are treating a patient. And that target or that particular gene or protein will some other day lead to its recurrence.
Thank you so much for your approach because it was so delight to have you on our platform to understand the concept. The first you have explained the biology behind it, the pathophysiology behind it, and then the disease which we do lack till today, and we need more number of research, more number of people who do research in cancer, and they can make it out someday that the kind of a treatment is available. So the caregiver burden, which is also responsible in cancer, and second, the patient burden in terms of pain, and pain is a major factor which a person go through during the process of cancer or the treatment of cancer. So hope that we all, the fraternity in the medicine, will work on this and we will help towards the treatment of cancer in the field of integrative medicine rather than only in the one system of medicine, because as we understand in the current era we need the understanding of all the system of medicine. So thank you so much for your kind approach to from start to end, and it
Thank
was a
you.
very short and sweet understanding of how we can understand the terms of research which we can improve to the treatment modalities in India. So thank you so much to come on our platform for this wonderful talk.
Thank you. Thank you so much.